start by asking how many of you are engineers, non-engineers? How many of you are engineers? Show of hands. Wow. Okay, so let me start with at least something that you guys, I'm sure, remember from when you were growing up. I remember it growing up. 1969, man walking on the moon. My dad was in the US and took a picture of it from a black and white television in his hotel. And it was the most prized possession that I grew up because it was a picture of man walking on the moon. And I'm sure you all grew up with that, right? Picture of man walking on the moon, featured in Time magazine. 40 years later, outer space had already been explored. We started exploring inner space. Unfortunately, Time magazine was really struggling with finding a great picture. Overdressed man on the moon, wonderful. So what do you do when you're exploring inner space? You put a picture of people underdressed and carefully censored by the double helix of DNA. That is what Time magazine did. But nevertheless, it was still a wonderful giant leap that mankind made. Small steps for man, giant leap for mankind, a phrase that has been immortalized by Neil Armstrong, could hold as much true in 1969 as in 2003. But it's post-lunch. I'm sure you're thinking, my God, we have to listen to biology. This is a reason I did not do biology after college, right? I went into engineering. So but I'm sure all of you are very competitive. So what I'm going to do is wake you up and boost all your competitive genes into overdrive with a little pop quiz, okay? Ready for it? Are you ready? Yes? Okay, if nothing else, at the end of the day, you'll have some amazing facts about biology which you can stump your kids with. Say, see, I know biology even though I'm an engineer. Okay. Okay, Morse code written in the alphabet of 2 letters, right? How many letters does DNA have? Oh my God, people, 4. Oh wow, you guys are great. You did biology also, wonderful. So what are those 4 letters? ATCG. My God, you guys are stumping me and I thought you'd be completely stumped by these questions. So AT and CNG, they create the double helix. About 1953, Watson and Crick discovered the structure of DNA. These 4 molecules basically make the basis of life from the time life began. Most living things have DNA in them and all of it is ATCNG, right? So great. Next question. Perfect. I see some people remember their biology very well. Okay, King James Bible contains 3 million characters. How many in our genome? Okay, you guys know biology, so I want to hear a few more answers. Any guesses? They already gave me the answer, so you can just repeat what they said. It's 3 billion bases. Okay, so many letters in the human genome. But how much of it is actually useful? Well, this is where the biologists start to hum and haw. There used to be an estimate of 50,000, so 2% of your genome. Now it's down to about 20,000 genes or so, and less than 2% of your genome is actually making the proteins that make us do everything, okay? So what about the rest? What is this 98.5% actually doing? Is it doing nothing? Where does it come from? Why does it even exist? These are some of the challenging questions that biologists are still grappling with. We're slowly discovering things about ourselves. So for example, we know now today that 8% of our genome is actually from viruses. All of you are familiar with HIV. It's something called a retrovirus. These are also retroviral DNA, which are there in our genome, which probably came from billions of years of evolution as man became infected with viruses. They stayed in our genome and are still there, okay? So we're trying to discover what these are, okay. How close are we to our nearest relative, the chimpanzee? By the way, this is not a chimpanzee, this is a bonobo, another group of the ape family we are very closely related to. How close are we to them? My God, you guys know the answers, 98%. How close are you to the person sitting next to you? Also 98%, hopefully a little bit more than that, just a little bit more, about 99% to 99.9%, depending upon your race, gender, ethnicity, etc. Okay. How unique are we, right? Name 3 things that impact our everyday life that came out of the space program. This is my favorite one, which is an infrared thermometer. I have a 3-year-old daughter. I'll talk more about her. But it's amazing because you can use an infrared thermometer instead of doing what my parents did, which was to stick a thermometer where the sun don't shine, right? So this is my favorite one. What about yours? You have two favorite ones? Sorry? Non-stick, Velcro, ball pen, lots of them, right? What about the Human Genome Project? What's come out of genome sequencing? What impacts your everyday life that has come out of genome sequencing? Okay, good question. So I will come back and ask this question at the end of the talk. At the end of the talk, I will ask this again. Okay. So today I'll talk a little bit about genomics, what they are doing, how they are transforming healthcare. Now that we've done this pop quiz and all of you did brilliantly, it's time to get personal, so we'll talk about healthcare, okay? This is India, okay? Lots of people. I was at CMC Vellore last week. There are people literally standing next to each other on the streets, on the roads, sitting on the ground, right? But India is also changing. 2000, just before the human genome got sequenced, not that there was any relationship, the biggest reason for what we call disability-adjusted life years, or things that affect the health of the nation, were neonatal preterm birth, lower respiratory infections, and diarrheal diseases, all infectious diseases. So the reds are the infectious diseases. 2013, we are becoming an affluent and an unhealthy nation. We've got heart disease and COPD as the top two, right? So what's happening? And what can genomics, if anything, can do for us to change this? Or will this always be, as we get more and more affluent as a nation, as we get more and more technology, digital, all of that, will we just be like every other country? Okay. So the whole idea when the Human Genome Project was done is that once you know all your genes and you know what they do, you'll be able to predict on a daily basis what is your risk for getting any disease, even starting with something as simple as heart disease, risk of getting stroke. Pharmacogenomics, tailored drugs, you'll be able to tell if a drug works today. Today, once you know you have a disease, the doctor prescribes a medicine, you take it, and you hope you will get well. If you don't get well, what do? You either change the medicine or you change the doctor. One of those two options, right? You go to the next hospital, right? So the idea was that this would all help because you would get better drugs, better treatment, better way of understanding your risk, better health, better wellness. Okay. So, but is it really going to happen, right? So to understand what biologists, scientists are trying to do when they look at genomics, when they look at genes, and how do they link to disease, right? So whenever we hear the word mutation, it's always this image of, for me, it's X-Men and Mutant Ninja Turtles, where the mutation gives you these amazing superpowers, right? One may argue, is it disease, is it mutation, right? A view of what autism is, and this is true. Autism is a disease, a genetic disorder, which you can diagnose while the child is in the womb. One view is, because autistic childs have sparks of geniuses, you are depriving the world of geniuses if you remove people who are autistic. On the other hand, for the parents who would have an autistic child, is it a hope? Now, to understand what these mutations are, how do they work, what are the biologists trying to distinguish? What does mutation mean? Any ideas? What is mutation? It's a change, right? So what kind of changes are these biologists and scientists looking for? So I'll come back and talk about my daughter now. Okay, this is my daughter who says, can you open the window? I want to see the hot guys. Okay, my daughter is all of 3 years old. So you can imagine my reaction when she said this. We were driving in a car. It was a hot Bangalore day. And this was her comment. So my reaction, okay. Next thing, my daughter, it is very hot. Okay, so now I understand, because I know what my daughter does, is she likes to use the word guys. And that's because I use the word guys a lot. And there's a very critical comma that was obviously not obvious when she said it, right? She wants to see the hot. It's very hot, okay? So mutations are like that. It's a single change in your bases, in your 3 billion bases, a single change that can happen, okay? A single change could be an addition, it could be a deletion, it could be whatever. And that single change makes a world of difference to what those words or those genes actually do or mean, okay? Sometimes the changes are longer, but for the most part, we're looking for the single change in a base, and that can change the meaning of what you look at, okay? Just to give you an idea, about 2012, it was in all the news, and I'm sure many of you have read this, is that people of the Indian subcontinent, especially India, are prone to heart disease, and it has been genetically proven. How many of you have heard that? Seen that news? So many of you, right? You believe it? You believe it? Why is there not a heart check that says, do you have that gene that tells you whether you're going to have heart disease? So I'll tell you the story about it. The story about it is, what was linked was a disease called cardiomyopathy. Cardiomyopathy basically means it's your cardiac muscles get affected and they do not pump as efficiently, okay? And so you're at increased risk for heart failure. There are 11 known genes in the world which cause cardiomyopathy. One of them, this one, the MYBPC3, sorry for the people at the back, the MYBPC3 gene is known to be more prevalent in the Indian subcontinent. In white is where you see the variation in the MYBPC3, and you can see the Indian subcontinent lighting up, and pink you see is not lighting up, right? So this gene is known to be different. Okay, now all of you engineers are going to love this. Great, so it is different. How many differences are there that make you different? 197. So if you want to design a sensor, okay, that detects whether you're at risk for heart disease, you've got to design a sensor that will pick up these 197 changes, okay, in your DNA, in the 3 billion bases that you have in all your cells, right? So that is what it will have to do, okay. By the way, the other genes are also known to be present. They're present across the world, okay? So if I'm trying to test for genetic predisposition, do you have risk for heart disease, right? You're going to have to test for all of these, and you have to test for every variation that could be present. Somebody talked about big data previously, right? So you have all this data coming in, and you have to interpret whether all of them combined give you an increased risk of heart disease. By the way, individually on their own, they don't increase your chances of getting heart disease possibly. But together they might, so you need to understand that. We talked about pharmacogenomics and getting better drugs, better medicine. Is this ever going to be a reality? Is there ever going to be a better drug? What we know today is at least whether there are ways of testing whether a drug will work. So I'll talk about a drug called warfarin. Warfarin is a blood thinner. It was originally developed as a rat poison and then shown that the reason it worked, it was thin the blood in rats, okay? Now, it became very useful as a medicine because obviously you need blood thinners for people who have heart disease. My mother is a heart patient, she takes blood thinners, okay? So it is the most prescribed drug in the world. It is on the essential drug list for every country of WHO, etc. Okay? This drug doesn't work equivalently for everyone. For a long time, it was believed that it was your gender and your ethnicity that determined whether you would respond to warfarin or not, okay? The trick with warfarin is if you take too much of it, you will bleed out because it's a blood thinner. If you take too little of it, it is not effective. So the question was, what is the right dose? And the dose seemed to change for people from person to person. So what people finally figured out is that there are genetic variants, CYP-C and VCOR, which actually tell you whether you will respond to warfarin or not. So today, if somebody is prescribing warfarin, the doctor is supposed to test your genetic profile. But I can tell you for a fact no one is doing that, okay? But he's supposed to do that, okay? Okay, great. So we had all this wonderful human genome project. Cost, I think, the US government, by all records, spent about $25 billion, not counting what Europe spent and all of that. And we know a few things after 12 years. And we know one thing, and I always quote Aldous Huxley here, who said, science is wonderful, it teaches us many things. Most of us, it teaches us what we don't know, right? So one of the things that we've learned along the way, we knew along the way, that genomics is not the answer to everything. The environment plays a big role, correct? All of you would agree? Great. I always show this picture. Is it nature or nurture, right? Two women of India, very well known, right? I do not have to name them, right? But is it nature or nurture that makes us who we are, right? So is it genetics, is it environment? And most people will answer, it's a mix of both. Great, wonderful. But then, if it is environment, do we even have a prayer of doing anything with genomics? because I can't control the environment. Or can I, right? So what is environment? And I think that is the next frontier that I want to talk about. From mice and men, the traditional approach of medicine, I think there's a new frontier that is emerging, probably for the last 2 years, of microbes and men. And I can tell you microbes are my new favorite things. Okay. Okay. Humans. 10% human, 90% microbial. Why do I say that? Because 10% of your cells are human cells. About 10 to 20 trillion cells that you have in your body are actually human cells. You have 100 trillion bacteria or microbes in your body. Okay? Together, they weigh about 2 kilos. This is wonderful news. So recently, when my friends and I were exchanging WhatsApp messages about weight loss and things like that, and people asked, what's your weight? I immediately subtracted 2 kilos, because that's the bacterial weight, right? 20,000 genes, 8 million genes floating around in our body, that's not ours. Are they doing anything? What are they doing? So about 3 years ago, they decided to sequence all the bacteria that are in our body. And the way you get at the bacteria in your body is you look at samples from the oral cavity, from your poop, from your pee, everything, right? Most disgusting, but that's how they do it. And the objective of that project was to find out what is there in our bodies and do they have any bearing on what we do, okay? And we're discovering a few things. I'll revisit heart disease. So we all know, we've all been told, if you eat red meat, you will get cholesterol and you will have heart disease, right? Correct? Yes? Okay. Okay, so how does it work? And why do I talk about microbes and red meat? What do they have to do with each other? So what's been understood now in a study that was done in mice, where they looked at mice, fed them a very high fatty diet, looked at their poop and decided how does it work. And they know that there's something called phosphatidylcholine, which is an ingredient of high fatty foods. The bacteria in your gut take that choline and convert it into something called TMA, trimethylamine, doesn't matter. And they convert it into a metabolite. Trimethylamine is a gas, so it moves to your liver. And from there, it becomes a metabolite that actually is implicated in forming the plaques in your body that contributes to, heart disease. Okay. So it was a study that got published in Nature. There was a lot of brouhaha. And people said, well, it's mice. So they repeated the study, and they showed that if you are vegetarian, right, and so what they did is they took about 75 people, and to those 75 people, they gave a pill every day, which mimics whatever is in red meat, okay? And they looked at what was happening with them, with the TMO levels, to show that whether they were getting increased levels of TMO, which would link to heart disease. So 26 of them happened to be vegetarian, and they did not see any increase in their TMO levels, so less likely to get heart disease. The omnivores, and I happen to be one of them, right, seem to have more TMO in them, and therefore more likely to respond to heart disease. So vegetarians, yay, right? Okay. Slightly more, so what they did then is, okay, is it really vegetarianism? Why is the gut flora different? So what they did is they actually went and looked at the gut flora, or they sequenced the gut microbes from the group. And they found that they actually did not for the whole lot, but about 50 of them, and found that for 4 of them, they have a species called Provotella of bacteria. And if you have Provotella, you actually get more of the TMO and more likely to get heart disease. Now, this is a very small study, but it shows up the possibility of saying that when we talk about environment, and there's no better example than saying, red meat links to heart disease as an environmental factor or a dietary factor, right? Your microbes are playing a role, and what microbes you have. By the way, there was an interesting follow-up study which they did where they looked at children from Italy, I hope no Italians here, who eat pasta, and they looked at children from Africa, and they found that Provotella was more prevalent in people who eat processed foods. Okay. The other thing which you might wonder is, where do we get our gut microbiome from? Where do all these microbes come from? When a baby is in its mother's womb, the baby is sterile. There's no microbes. Okay, the mother's placenta protects it. Okay. The first time that the baby acquires microbes is as it is being born, if it is born by a natural birth. through the vaginal canal, okay? Over the next 3 years, the first 3 years of life, the microbe environment in the baby's system builds up and becomes like that of an adult, okay? So if you really have to impact your kid, you have to do it in those 3 years to make sure they get the right microbes, right? Unfortunately, my daughter is already 3 years old, so I'm past that, okay? But that's what you have to do. So what if you know that there are microbes which affect your gut flora, right? And I've talked about heart disease, but I'll hark back to infectious disease in a minute. Because the first thing people said is, well, if I know that I can change the gut flora, then maybe there is a way of me controlling what disease I get, right? So there are 13 companies already in the US. Since we knew first about microbial genomes in 2012, 13 companies have started up in the US which are trying to build drugs based on microbes, okay, or targeting the microbiome, okay. Many of them are probiotics, so I'm sure you've seen the ads. Yakult and others, right? But don't start going and drinking Yakult because I said so, okay? I'm not advertising for Yakult. But nevertheless, the idea is that if you can get the right set of microbes into your body, maybe it will help you, okay? This is unproven, but we are just at the brink of it. And the reason I want to talk about that, even though we're on the brink of something, it is still at the edge of science, we're still trying to prove things, is because of an actual experience that we are going through. Right. In my company, we're working with the Satyasai Hospital, and one of the most common infections that they see is urinary tract infections, especially with drug-resistant E. coli. Now, WHO lists drug-resistant E. coli as the most common and the most difficult to treat bacteria today. The view on antibiotic resistance is probably the most grim. Even though we are starting to see more heart disease and less infections, this could all go away and come back with a bang if antibiotic resistance doesn't get treated well, and we're probably on the brink of an epidemic, right? So people talk about antibiotic resistance. You see items in the news about superbugs, and how do you treat that? The difficulty with antibiotic resistance is that there's no pharma company making new antibiotics, okay? So if you are resistant to an antibiotic today, you probably have no choices left. So people are starting to look at things like the microbiome to see if they could treat drug-resistant infections. Okay. So I think one hope for us in all of this microbiome project is to find out new medicines that would treat things like antibiotic infections. Okay. I talked a little bit about heart disease. I talked about infectious diseases at the very end. And I want to just say this about infectious diseases, is that they are neglected. Neglected because of the social and cost implications that it holds for most companies, okay? Just to give you an idea, I've talked about genomics, and you're probably thinking genomics, testing for genes, this is like the Ferrari of medicine, right? This is like premium medicine being offered to the very few who can afford it. The cost of genome sequencing has come down in the last 20 years from about 100 million to about $1,000. But still, $1,000 is outside the realm of many of the people in India who go to places like this. So we have a motto. We talk about make in India. We say heal in India, right? And you have to look at infectious diseases and the burden that it is causing, right? And what we believe is that if you can build technology that you can look at infectious diseases using genomics, you have a better hope of beating antibiotic resistance. So now I'll come back to engineering for all of you engineers, right? So the technology exists in big laboratories, right, to do this. There's nothing new in the technology. So what are the things that we have? Look at the specs. Your patient can pay about 10 rupees. Your customer is the doctor, and if I will say doctor within quotes, because many of them don't have an MBBS. If they do, they've never heard of DNA sometimes, right? So those are our customers, right? And you're taking something like a Ferrari and you're building a nano, if I may use that analogy, right? And you're trying to say that how do I do that at such low cost? So it's engineering of saying that I'm going to take something that can be done in a very beautiful, well-equipped laboratory and start to remove cost out of it. Start to miniaturize it so that the cost of my reagents come down, right? Start to look at, I heard about the EHS thing, right? Start to look at things where, the person who is going to the clinic probably doesn't have someone who can even draw a blood sample from them. So how do you handle that, right? How do you engineer a product so that it does not require anything to be either cold stored or to be heated when you're doing the test, right? Most laboratory equipment or most laboratory tests either require 4 degrees, -20, etc., or they require heating, if not both, right? So it becomes engineering of saying that you take something and start to build technology on top of it that will allow you to deploy it to the people who need the most. So with that, let me say thank you and open it up for any questions that you have.